closely connected. And today I'll be sharing two closely connected pieces of work. The first part of my presentation focuses on my master's research, where we developed environmentally friendly silver nanoparticles loaded with berberine to improve light-based treatment of breast cancer. And then in the second part, I'll broaden the discussion to look at the future of this field by introducing my recently published review on how artificial intelligence is beginning to transform cancer theranostics and precision oncology. Although these may sound like two separate topics, they are actually part of the same journey. My experimental work demonstrates what is possible with nanotechnology today, while the review explores how artificial intelligence may make these therapies even smarter in the future. So whether you come from medicine, biology, agriculture, engineering, computer science, business, or even if you have little scientific background, I hope you will leave today with a better appreciation of how different disciplines are coming together to improve cancer care. So you can see on my screen, let me use a pointer. Great. So my master's work was on the topic combined chemophototoxic efficacy of berberine-loaded green-silver nanoparticles against breast cancer cells. And this project was under the supervision of Prof. Blasan George, and then it was co-supervised by Dr. Paromita Sabadikari. And we all belong to the Laser Research Center from the Faculty of Health Science, University of Johannesburg. So the keywords I would want you to notice today and precision oncology. And these concepts will connect throughout the presentation. So before discussing our approach, let's first remind ourselves why breast cancer continues to be such an important global health challenge. So breast cancer remains the most commonly diagnosed cancer among women worldwide. Despite remarkable advances in medicine, millions of women continue to be diagnosed every year. And unfortunately, many still lose their lives to the disease. So one statistic that often surprises people is that approximately 1 in every 8 women will develop breast cancer during her lifetime. Now, it's important to recognize that tremendous progress has already been made. Earlier diagnosis, improved surgery, chemotherapies, the immunotherapies, radiotherapies, hormonal therapies, all these have contributed to increased survival. However, these treatments are not perfect. Just as we always say, efficiency of a machine can never be 100%. So many patients still experience significant side effects, treatment resistance, and then reoccurrences. So this raises an important question. Can we develop treatment that more precisely destroy cancer cells while causing less damage to healthy tissues? So let's just think of it. Cancer treatment, like trying to remove weeds from beautiful garden. So you have a beautiful garden, right? And then there are weeds amongst them, all right? And you are using like a powerful herbicide, okay? So while the weeds may die, many healthy flowers are also likely to be damaged, all right? So our goal is to develop treatments that behave more like carefully removing only the weeds while leaving the flowers untouched. So this naturally brings us to the limitations of the treatment we currently use and why researchers are looking for new alternatives. So current breast cancer treatments usually involve surgery, like I mentioned earlier, chemotherapy, radiotherapy, and all the others. One or many of them combined, all right? And these treatments have undoubtedly saved millions of lives. However, they also have important limitations, as I've mentioned already. For instance, surgery may not completely eliminate microscopic disease, all right? And then chemotherapy travels throughout the body, meaning healthy cells may also be affected, leading to familiar side effects that we all know, like hair losses, nausea, fatigue, and etc. All right? Radiotherapy is highly effective, but also can damage nearby healthy tissues. So in addition, some cancers eventually become resistant to treatment, meaning the same therapy becomes less effective over time. So these challenges encourage researchers like myself to develop more selective treatment. One promising approach is photodynamic therapy, where light activates a drug only at the tumor site. So in our work, we sought to further improve this strategy by watering an entire field because only one tree needs water. Just imagine, just one tree needs water, but you end up watering an entire field. That's exactly how conventional therapies sometimes work in a similar way. So precision therapy aims to deliver treatment exactly where it is needed. So if you look at our goal here, our approach is actually using photodynamic therapy, green synthesized silver nanoparticles that is loaded with a photosensitizer. I would explain. So this schematic diagram speaks just it's just an overview, right? So there's going to be a plant extract somewhere. There's going to be green synthesis. There's going to be silver nanoparticles being formed. PS here is photosensitizer that has been loaded onto our synthesized silver nanoparticle. And then there's eventually going to be a laser that has been thrown on it. I'll give proper context and proper explanation. So the greater goal now is we want improved stability, enhanced biocompatibility, and ROS, which is reactive oxygen species generation to kill cancer cells. One lethal or one compound or things that really kill cancer cells are reactive oxygen species. All right. So you'll be seeing this ROS very frequently. So to understand our approach, we just need to also understand how photodynamic therapy actually works. So let me just quickly mention on what PDT or photodynamic therapy does. All right, so it's actually a remarkably elegant treatment. It relies on three essential ingredients, okay? The first is a photosensitizer or PS, a molecule that responds to light. And then the second is a specific wavelength of light. And then the third is molecular oxygen, which is naturally present in our tissues. So I would use this scheme to explain what we are trying to discuss here. So you have a tumor that is vascularized, so there's the veins, blood running down and up and down. Okay, so using EPR mechanism, your photosensitizer will move into your tumor and it will be accumulated. And once it's accumulated in the tumor, we throw light on it or laser light at a specific wavelength on it. And then this photosensitizer, as the name implies, it's a photosensitizer, which means it has the ability to absorb the light from the laser and then generate what we call reactive oxygen species. So that is has been summed up here. So your PS has been excited because it came in contact with the light, right? HV. And then it produced these guys which are lethal to our cancer cells. So eventually we see apoptosis or necrosis. So a useful way to think about PDT is to imagine a glow stick, okay? So before you bend it, nothing happens. But once it's activated, it produces light. Similarly, a photosensitizer remains relatively inactive until illuminated with a correct wavelength. Sorry. So many photosensitizers exist, but we became very much interested in naturally occurring compounds because of their excellent safety profile. That is why you heard me saying earlier on green nanotechnology. Green nanotechnology. It means there are chemical ways of also making the nanoparticles, but we chose to focus on green because of safety profile. It is environmentally friendly. We're trying our best to move away from harsh chemicals in forming these nanoparticles. So to touch on the phytochemicals in breast cancer prevention and treatment, the next question is which molecule should be used as a photosensitizer? Okay, so researchers have developed many synthetic photosensitizers, as I mentioned earlier, over the years, and some are already used clinically. Okay, however, nature also provides us a remarkable source of biologically active compounds. Many medicinal plants contain molecules that have been used for centuries in traditional medicine. and are now being scientifically investigated for their anti-cancer properties. So these natural compounds, many of them I have listed here. So you see berberine and then chlorophyll derivatives, isoflavonoids, and the likes. Okay, they come with many advantages that I've mentioned here. Cell death, they enhance cell death. There's pro-oxidant, they're antioxidant, they have less side effects because they are natural, and many others. Okay, so all of these contribute to cancer prevention and treatment. But like we always say, efficiency of a machine can never be 100%. So we've also realized that these compounds that are natural photosensitizers come with some limitations. Okay, they have poor bioavailability, water insolubility, low cell penetration, very narrow therapeutic window, unfavorable pharmacokinetic parameters, and higher hepatic disposition. So let's to make everyone to understand what I'm trying to say, because in the audience we have mixed audience, right? We have people who are not really inclined with cancer research, right? So think of these natural compounds as a talented athlete, okay? They have tremendous ability, but if they cannot get onto the playing field, obviously they cannot perform, all right? So in the same way, many plant compounds have excellent anti-cancer potential, but in a struggle to reach to the tumor in sufficient amounts because of their size and also bioincompatibility. All right. So one of the compounds that attracted our attention was berberine, which became the center of this research. So berberine is naturally occurring. It's an alkaloid. It's found in many medicinal plants and it has received considerable attention for exhibiting multiple anti-cancer activities. Studies have shown that it has increased oxidative stress within cancer cells. All right. So it triggers apoptosis. It also suppresses stemness-associated pathways. So these are the advantages of berberine. It's a really good, it has serious anti-cancer properties. But then like many other phytochemicals, which are photosensitizers, it comes with its own limitations like poor water solubility, poor bioavailability, unstable, and has low cell penetration. And then also you require very high doses for you to see a significant therapeutic index. So then the question led directly to the rationale of the study. Okay, so at this stage we had two important observations. Okay, first, berberine possesses excellent anti-cancer activity, but suffers from poor delivery. So this is berberine. So then, how do we improve berberine's mobility? Okay, so that is where we introduce what we call the silver nanoparticles. And it is green because we use Sina didymobotria, which is a plant, to do the synthesis. Remember, I mentioned that we're trying to play safe because of environmental issues. So instead of us to go for harsh chemicals for synthesis, we are rather relying heavily on plants because of safety. So we use the plant to form a silver nanoparticle. So now it becomes a green silver nanoparticle. So if berberine has a problem of being delivered to tumor site, then let's enhance its solubility by conjugating this berberine on our silver nanoparticle. And of course, it involves some chemistries. So we've realized that um, in addition, if you put them together, we expect combination of improved stability and also increased cellular uptake, enhance reactive oxygen species production during photodynamic therapy. And also, we want to produce greater cancer cell death than either component alone. Silver nanoparticle on its own, studies have shown that it has lethality against cancer cells. And there's one article that I published from this whole master study that I compared chemically synthesized to green synthesized. And you can see from that side, all right? So silver on its own has anti-cancer, berberine on its own has anti-cancer, but the study here is synergistic effect. If you can improve the delivery of berberine by coating it on this, and both of them can give us anti-cancer, then hooray, it's a win-win. So that was the rationale. So we conjugated so that we decrease the delivery dose of berberine and silver, and then also we heighten our synergistic or additive anti-cancer properties. So the overall aim of this work was straightforward: to develop environmentally friendly berberine-loaded silver nanoparticles and determine whether combining the chemophototoxic therapy could improve the treatment of breast cancer cells. And to achieve this, we had to stratify the main objective into 5 specific objectives. So we synthesized green silver nanoparticles. So I use green because we use the plant Sina didymobotria, just so that you understand. We also synthesize stable BBR AGNPS. So this is a conjugate. BBR stands for our berberine, and remember AGNPS is our silver nanoparticle. So this is like joining them together. BBR, AG, and PS using eco-friendly methods. I already mentioned we are using a plant, so it's eco-friendly. And also we characterized the uncoated, which means that there were some silver nanoparticles that we did not conjugate. We did not put the berberine on top of it, right? And then there was one that we put berberine on top of it so that we need to understand if there is berberine attached, what is the effect? If there's no berberine attached, what is the effect? And characterizing means that we are looking at spectroscopic and microscopic appearances. So we need to define how the sizes are looking and etc. I'll explain further as we move forward. And then we also assess the combined and individual effects of uncoated and then the conjugate against our breast cancer cells using cell-based biochemical and molecular biological assays. And also we evaluated the potential of both the nanoparticles to induce cell death pathways in the breast cancer cells through chemotoxic and phototoxic effects. You would appreciate these very soon. So rather than focusing on every experimental technique, I would rather like to walk you through the overall workflow. So the study was actually stratified into 4 methodologies, literally. So first, we cultured 2 breast cancer cell lines. We used MCF7 and MDA-MB231. These are breast cancer cell lines. And then which represent different forms of breast cancers, all right? And then 2, we synthesize the silver nanoparticles using plant extract. So this is more like a scheme of what was happening. So you have a plant and you dry it, you dry the leaves. We use the leaves because we need most of the phytochemicals. So we use the leaves, which are reducing. So we use the leaves. We dried it under the sun, and then there's some maceration. I think there are details in the slides. There was some macerations, and then we did extraction. And from there, we had silver nitrates, and then the extract. We put them together, give a certain condition, and then eventually we form our silver nanoparticles, and then we coated it with our berberine. And then, like I said, characterization, which means we need to understand what is happening. We want to know the size. So all these guys listed here, the UV-Vis, FTIR, DLS, TEM, SEM, you would understand them as we move forward. But then understand that they are all trying to let us understand the charges on our nanoparticle, how the nanoparticle looks like, and the elemental components, all right? And then also we did stability studies because everyone would understand that if you have a nanoparticle, it should be able to carry the berberine, but it should be able to also to be stable enough. Yeah, it should be stable enough if you introduce it into the tumor. Okay, so we did all these studies, which I'll explain later on. And then thirdly, we exposed the treated cells to laser light to activate the photodynamic process. So I used 405 nanometer laser. So it was thrown onto the cells, the breast cancer cells that has been treated with our nanoparticles. So this is a dose response study, and you would understand later on. And then eventually we did evaluate berberine coated 7 nanoparticles, how it induces cell death. Okay, so these are major assays that we looked at: MTT assay, morphological analysis, live death, etc. You would understand as we move on. So having established the methodology, let's now move into the results and see what was going on. And the first step is actually to ensure that we were working with the right or the correct plant species. So the leaves, we collected the leaves from Limpopo province here in South Africa, and then we formally authenticated it because we submitted it to a botanist, and then there was authentication. And then a voucher specimen was then deposited at the university's herbarium. So as you speak, we still have it there. So this may seem like a small detail, but it is actually essential for scientific reproducibility. So if another research group wants to repeat our work in future, they must begin with exactly the same plant species, all right? Proper authentication ensures that our findings can be verified and built upon by other researchers. Okay, so once we have confirmed the plant material, we can now move into synthesizing our nanoparticles. So briefly, this is what was going on. These are live pictures that I took of the process of forming our green synthesized silver nanoparticles. And I'm going to walk you through quickly. So like I mentioned earlier, there was a plant, we dried, macerated, and then we took 10 grams of it in 100 millicube water. And then there was an extraction, and then we got our plant extract from it. And then now there was a mixture of plant extract and silver nitrate prior to stirring. So this is how it looked like. All right, if you put the plant and silver together, see the color. And green silver nanoparticles after 24-hour vigorous stirring. So the moment you see this color change after 24 hours, it means your silver nanoparticles have been formed. So we are going to deal with this. And then also remember, we need to conjugate it with berberine, free berberine. So free berberine has its own color, as you can see here. If you add it to this and then give it the right conditions, eventually you form your conjugate, and this is how it looks like. Okay. So color is only the first piece of evidence in forming your silver nanoparticles. But then we need analytical techniques to actually confirm whether, okay, what we are saying is actually berberine, whatever, and then green silver nanoparticles. All right, there's an important thing I needed to mention. This reducing potential of leaf extract was actually done prior to the silver nanoparticle synthesis because we needed to know the reducing potential of our leaf extract. Okay, because remember we are reducing the silver nitrate into silver nanoparticle. That's the chemistry here. So we want to rely on the reducing agents or the reducing compounds in the plant rather than rely on chemicals out there that can help us reduce. Remember, we are moving away from harsh chemicals. Okay, so safety profile. So plants have reducing, some plants do have reducing properties. So we needed to measure. So the reducing potential of the S-didymobotrial leaf extract was calculated, and we found it to be 9.58 plus or minus 0.12 milligram per the gallic acid extract equivalent, all right? So per gram of dry weight, okay. So one of the first techniques that we used was the UV visible spectroscopy, and that is this. So quickly, if you can see clearly, we have free berberine, which is then a cyan color, and then we have green silver nanoparticles giving us this, and then we have berberine, the conjugate, giving us that. I would explain. So what is happening here is when you have the plant alone, which is the brown thing, you see the absorbance just went down. There was nothing in here. This side is just noise. The plant is the brown one, right? There was just noise. That was just the plant extract. Okay. And then we have our green silver nanoparticle. That is when we use the plant to synthesize the silver nanoparticles. It gave us this. So we had an absorption of 466 nanometers, which is this one. Okay. So it is communicating to us, okay, there's something there. Because if the plant alone is this, and then after using the plant to form the silver nanoparticles, it's giving you this. So it's a bit confirming to you that, okay, there is the formation of what? Silver nanoparticles. So the strong SPR, which is surface plasmon resonance, which is a bit technical, it was 466 nanometers, confirming nanoparticle formation. And then our conjugate exhibited a peak at 467. So if you look at berberine, silver nanoparticles, the conjugate, it's this and that. So the peak is 353 here, and then there's another peak 467. Now you can confirm to some extent that there is conjugation because of this peak, which is 467, which is closely related to the green silver nanoparticle. So you can agree that after adding the berberine, there was some formation. Because if you look at just the free berberine alone, it has two peaks, 345 and then 422. But now there's a shift in the peak, moving towards to 467 after we added the two together, both the green silver nanoparticles and the free berberine. So that is also confirmatory enough for you to like, okay, be like, okay, have confidence and say, yeah, we are getting there. But then we need more of these analytical methods to actually be very sure what we are dealing with. So that brings us to this slide. So the next question was whether they possess suitable physical properties for biological applications. So we did FTIR spectroscopy, which actually showed natural compounds from the plant extract, which were involved in reducing and stabilizing the nanoparticles. And it's a bit smaller, and this data will be available very soon because this paper has been submitted for publication. So I'm looking forward to it. Most of us would appreciate it when we chance on it and we read. You find the details of the methodologies and the explanations and probably the visibility. I'm sure most of you are struggling to see. So, but then the greater good here is that we were able to identify certain compounds that were confirming to us, okay, our conjugates were formed and it was there. Okay. And then we also did something called particle size and zeta potential analysis. What this English is trying to tell us is the charges on our nanoparticles. We needed to know the charges on our nanoparticles. Okay, so as expected, the nanoparticles became slightly larger after berberine was loaded. Okay, after berberine was loaded onto the silver nanoparticles, the size became larger. And then the final charge became very negative, which is an indication that the nanoparticles was collidally stable. So they were unlikely to aggregate excessively during storage if you store them or in biological experiment, all right, which is a good thing because remember, like the silver nanoparticles would encounter in so many physiological conditions. So if you put them in a physiological condition and it doesn't work, there's no use of it. Like there's no use of it. So the next, we examined what these nanoparticles actually looked like under the microscope. And this is what we found. So we did what we call surface morphology and elemental composition. That's using SEM, which you saw earlier on. I told you, like, as we move forward, you would understand these terminologies. So there were two microscopes that were used, the scanning electron and then this the transmission. So this was just to look at the surface of our nanoparticles. All right, so green silver nanoparticles showed irregular but well dispersed, while the conjugate were more clustered, indicating surface coating by berberine. So you can see amongst the two. This is just the green silver nanoparticles, and when it was coated, you can see the clustering. And then we did EDS, which confirms silver. So this is the EDS, the elemental mapping. This green synthesis has its elemental mapping here, and then the conjugate also has its elemental mapping. So the elemental mapping literally is telling you the element present. So we found silver dominating here. We also found silver here, which is an indication that the silver nitrate that we use to form the nanoparticle is present in the final nanoparticle that we form. So it also confirms for us, and we have some level of assurance that it is there. Okay. And then the term actually measures the size, the actual size. So the term images show that there's green silver nanoparticle were generally spherical and were dispersed, which you can see this image, the A. Okay. And it was averaging around 39 nanometers. After loading with berberine, the nanoparticle became slightly larger. So this is when the berberine has been coated on, and we took the image under the microscope. And we were now the size now was now 57 nanometers. So it's also an indication that okay, something has been coated on our green silver nanoparticle. All right. So producing nanoparticles is only useful if they remain stable under storage and biological conditions, which I've explained earlier. So all these are just telling us, okay, are my things stable? Okay, are my things stable? Because of time, I wouldn't be able to explain each graph that you see. But then we evaluated the stability under storage conditions and then under conditions that mimic the human body. So encouragingly, both nanoparticle formulations retain their characteristic optical properties throughout the study. And importantly, the berberine-loaded nanoparticles remained considerably more stable than the free one. So our conjugate was more stable than the free one. And like I said, time will not permit you. I would have shown you. I would have shown you. So this is important because a therapeutic formulation must remain stable long enough to reach its targets before it's being what released. This slide evaluates the performance of the nanoparticle as drug delivery system. Remember, we said berberine was having poor delivery, so we needed to coat it with to coat it on top of a silver nanoparticle. Okay, so we needed to now measure the entrapment, how much of the berberine has been loaded. Okay, that's just this information. That's what it's presenting. All right, so encapsulation efficiency we measured was 90%. Okay, which is like 90% of the berberine used during the preparation became successfully encapsulated within the nanoparticles. And it tells us that very little of the drug was wasted during the formulation. So during the time that we were forming our berberine silver nanoparticles, we lost about 10%, but we managed to load 90% of it, which is good. So we observed an initial rapid release during the first 2 hours. So this is a release study. We observed an initial rapid release during the first 2 hours, followed by a slower sustained release over the next 24 hours. Okay, so this type of release profile is often desirable because it provides an initial therapeutic effect while maintaining drug availability over time. Okay, so finally, we investigated whether the nanoparticles responded to light upon laser irradiation. The berberine-loaded nanoparticle produce a greater temperature rise. So let me quickly, this slide. There's another term that has been introduced here, which is called photothermal activity. Just like photodynamic therapy, photothermal does not need a photosensitizer. It literally relies on just the delivery mechanism, which in this case is silver nanoparticle. So we wanted to see if we throw the light or the laser on the nanoparticle, it will be able to absorb the light and produce heat, because photothermal, as the name implies, using light to generate heat, thermal. So we need the heat to kill the cancer cells. That study is on its own, all right? But we just wanted to see. And at the end of the day, we appreciated, if you look at A and B, this is water at 0, and we were reading temperature of around 12.1 degrees. And when, after 5 minutes, right, the temperature was still around 12.8 degrees Celsius. So there wasn't any significant rise, okay, because it's just water, it's a control. And then C and D, are green silver nanoparticles. So this is green silver nanoparticles at 0 and after 5. Okay. So you realize initially it was around 19.3 degrees, and then after 5 minutes there was a temperature rise, which is around 20.1 degrees Celsius. So it means the nanoparticle was able to absorb the laser and generate some temperature, which it's not really significant because that's very minimally small. It's very small. It's very, very small. So, and then the last, this is the conjugate, all right? The conjugate under 0 and the conjugate after 5 minutes when you irradiate, okay? There was also a temperature rise. So there was approximately 2.8 degrees Celsius rise. So the green silver nanoparticle showed minimal temperature change, which was very close to water, indicating it had a limited photothermal efficiency without berberine. Yeah, just to add on, I mentioned earlier something called reactive oxygen species. And this is key because it's necessary to kill, we need it. We actually need it to kill the cancer cells. We need to generate ROS as long as photodynamic therapy is concerned. So this is the data we generated when we measured the ROS generation and singlet oxygen. Singlet oxygen is also lethal to cancer cells. So not to bore you much, literally the combined system, which is the berberine silver nanoparticle, gave us an overall very, very good reactive oxygen species. We were averaging around, the FT to FT naught was around 1.6 and 210 for both the singlet oxygen and reactive oxygen species generation, which is good, which means that our conjugate, when you introduce into the tumor, it would be able to generate that amount of reactive oxygen species to kill the cancer cells. Literally, that's what it means. Yeah, there's a lot also on this. This is in vitro anti-cancer activity, looking at the chemo and phototoxic effect. Basically, we were treating the cells, the two cell lines that I indicated earlier, the MCF and the MDA. So we treated those two cell lines with the silver nanoparticles that was green synthesized, and then the conjugate berberine silver nanoparticles, and then also free berberine. Okay, so we wanted to see if you treat the cells with free berberine, what happens? If you treat it with the conjugate, what happens? And if you treat the cells with just the silver nanoparticles also, what happens? And like I said, there's a lot going on. There are some figures here which were very, very impressive. But then overall, we observed that the MCF7 cells were slightly more sensitive than the MDA, which means the MCF7 cells, like they were dying more as compared to the MDA, all right? And this is unexpected because these cell lines differ biologically, with MDA MB231 generally being the more aggressive subtype. So to some extent, it makes sense why it would show less effect on it, right? Yeah, so this slide also explains why the berberine-loaded nanoparticles performed so well. Rather than looking only at whether the cells died, we wanted to also understand what happened inside the cell before death occurred. Okay, so more like the cell death mechanism. Okay, we found that significantly more berberine entered the cancer cells when it was delivered using nanoparticle than when it was administered as free berberine. Remember, we were dealing with groups. So there was a group that was just free berberine, and there was a group that was the conjugate, and there was a group that was silver nanoparticle. And we treated the cells with all these groups. Okay. So we found out that there were more berberine that entered the cancer cells when it was coated around the silver nanoparticle. In this case, it means the conjugate was performing far better than when berberine was just alone, which confirms what we earlier presented, where berberine has poor solubility and biocompatibility and mobility. Okay, so it is important because a drug cannot work effectively if it never reaches its target. Okay, so basically that's what is going on here. This A and B is cellular uptake. C and D represent relative ROS generation in the cell. The earlier ROS or reactive oxygen species that I demonstrated was outside the cell, that was extracellular, it was cell-free system. We wanted to have an idea how much of the ROS can be generated if it is not in the cell. And if it gets absorbed into the cell, how much ROS is being generated as well. Okay, so this is the data that we generated, not to bore you much. And then this is the mitochondrial membrane potential. There was a significant loss of mitochondrial membrane potential. It's an indication that the mitochondria were no longer functioning normally, which means the cells were dying. Remember, the mitochondria is the powerhouse, okay, of the cell. So once the mitochondria fail, the cell begins an irreversible pathway that moves straight towards programmed cell death, and that's what we call apoptosis. So the disruption of mitochondrial function suggested that the cells were definitely undergoing apoptosis, and which were investigated in the next experiment. So this is just the apoptosis thing that we were talking about. The scatter plots transform into these nice graphs here. And what we are trying to say here is unlike necrosis, which is uncontrolled and can trigger inflammation, apoptosis is much cleaner and more regulated. So our cells were dying using the apoptotic pathway rather than just necrosis. That's what basically we are saying. Okay. So these findings were also reflected in the physical appearance of the cells. So let's look at the cells. Great. So A, D, G, H are control cells. So these are the cells when you've not treated them. They look very happy and excited. But then when you introduce your groups, okay, I just want us to appreciate what happens. When you introduce the groups, now you see the cells are not happy. They are shrinking, they are becoming more roundish. This is an indication that they are dying. B and C are different conditions. So B and H is berberine silver nanoparticles at an IC50 dark, which means there was no laser. IC50 means inhibitory concentration at 50. So we needed 50 a certain concentration to kill 50% of the cells. That's literally the meaning of IC50. Okay. So not to bore you much, this is just to let you appreciate that whatever I've been talking about, actually you could see them also. Because if the cells in the control form are not dying, they look happy, and you treat them, this is how they look stressed and they are roundish and all of that. It means our nanoparticles were working, and when we threw light on it, it was even worse. All these groups here is when they were treated with light. You could see it's even dying way faster. So I would conclude the first part of the presentation. So we successfully developed our environmentally friendly nanoparticle platform using our plant. And these nanoparticles, we were able to encapsulate, do the characterizations, and also we found the ROS generation ability. So overall, the combination of the green nanotechnology and photodynamic therapy produced a stronger anti-cancer effect than either approach alone. So the next important step will be to evaluate this nano platform in animal models or even 3D. I forgot to mention this study was done in 2D cancer cells, not 3D, which means the cells were grown on in a two plane like X and Y on a flat surface. Okay, but that is currently what I'm doing. My PhD now is moving from the 2D into a 3D. So 3D actually mimics the tumor itself. It's big, it's roundish, it's spherical. So we are dealing with spheroids. So we want to see how the cells will be able to penetrate a tumor, not just a planar. Okay. And everything that has been said so far, the schematic representation of the study is what is being shown here. Right, so from bench research to the future of precision oncology. Okay, our work shows that we can actually design smarter nanoparticle systems to improve light-based cancer therapy. But this naturally raises another question, like even if we develop highly effective nanomedicines, how do we determine the right patient, the right treatment, the right dose, and the right timing? So these are questions that we were asking. And these are increasingly complex decisions that cannot always be answered by just biology alone. So this is where artificial intelligence is beginning to reshape the future of cancer theranostics. So in the second part of this presentation, I would like to explore how AI is actually helping move light-based cancer therapy toward truly personalized precision oncology. So I actually published a review together with my team. So we published a review, and this is it. You can, at your own free time, search for it and then go read in detail what actually was mentioned. So the question, the word theranostics, what does it mean? Okay, so it's actually a combine or a combination of two words. It's therapy and diagnostics. Traditionally, diagnosis and treatment have been separate steps. Okay, so first, like a doctor will diagnose the disease, then they decide on a treatment, then they wait to see whether it works. Okay, now theranostics, it changes this model. It combines diagnosis, treatment, and monitoring into one integrated process. So instead of treating first and evaluating later, now the theranostic system is it's a continuous, so it continuously collects information during the treatment. So this allows clinicians to adjust therapy based on how the tumor is actually responding. The result is treatment that is more precise, more personalized, and potentially associated with fewer side effects because it replaces one-size-fits-all oncology with adaptive response aware treatment pathways. Right, I've already mentioned this. Our experimental work, it demonstrates that light-based nanomedicine can effectively destroy cancer cells. We've already seen that. So the next challenge is making these therapies smarter using artificial intelligence to determine the right patient, the dose, and the timing and treatment responses. So this figure that you see actually illustrates how theranostics recognizes the entire cancer care pathway. Instead of thinking of diagnosis and treatment as isolated events, we can think of them as a continuous cycle. Okay, so it begins with detecting the tumor. So once it has been identified, there's going to be light responsive agent which will accumulate within the tumor. So I've spoken about this in my previous submission, the first part of the presentation, where there was targeting, then the conjugate or the nanoparticles will be accumulated. You throw laser light on it. So this is the treatment part. And this is the part where AI can be introduced or is being introduced. Okay. So optical imaging then guides therapy while simultaneously monitoring the response. So based on this feedback, treatment can be modified in real time if necessary. Okay. So this continuous flow of information allows clinicians to make more informed decisions throughout the patient's treatment journey. The key idea is that every stage informs the next through data-rich feedback. Maybe there's too much right now, like to comprehend. So let me make it simple. Like think of a modern smartwatch. Okay, most of us do have smartwatches. It doesn't just count your steps once a day. It continuously monitors your heart rate, the activity, sleep, constantly updating the information, right? So diagnostics follow the same principle by continuously monitoring the disease during treatment. Okay, so it can actually inform the clinicians at any time because it's real-time monitoring. So if photodynamic therapy is so promising, why do we still need artificial intelligence? And then the answer actually lies in the complexity of cancer biology. Okay. No two tumors are exactly the same. Even within a single tumor, different regions may have different oxygen levels, blood supply, and photosensitizer uptake. So this means that applying exactly the same treatment protocol to every patient is unlikely to produce identical results, okay? And that's another challenge that befalls PDT. So light does not penetrate deeply into human tissue, limiting the treatment of deep-seated tumors, okay? Finally, many treatment protocols remain relatively static. The same light dose may be delivered regardless of how the tumor responds during therapy. And these challenges highlight the need for systems that are capable of actually adapting treatment in real time. And this is precisely where artificial intelligence begins to add value. Okay. Many people think of artificial intelligence as a replacement for doctors, and that is not the goal. Okay. Instead, AI functions as a powerful decision support tool. So it can rapidly analyze enormous amounts of information that would be impossible for any individual clinician to process manually. So at the detection stage, AI improves image quality, identifies subtle tumor features, and even during diagnosis, deep learning algorithms assist in tumor classification and segmentation. AI can then support treatment planning by predicting the most appropriate photosensitizer dose, light intensity, and treatment timing. So during therapy, continuous imaging allows AI to monitor treatment response and recommend adjustments if necessary. And so finally, after treatment, AI can continue monitoring the patient to help detect recurrence or predict long-term outcomes. So rather than replacing with clinicians, AI actually extends the ability to interpret increasingly complex information. Let's think of AI as an experienced co-pilot, okay? Assuming I'm the pilot and then AI is the co-pilot. So the pilot is flying the aircraft, but the co-pilot constantly monitors hundreds of instruments, identifies potential problems, and provides recommendations. So then now the onus or the final decision actually lies on me, the pilot. And that's a similar way when it comes to diagnostics. Clinicians remain responsible for patient care while AI provides additional insight. Okay. So you may be wondering whether this is still a vision for the future or whether AI is already making a difference. Well, the answer is that significant progress has already been made, and it has been illustrated in my next slide here. Um, we often hear that artificial intelligence is going to change medicine in the future. But the reality is that this transformation has already begun. Okay. The timeline on this slide, I don't know why my image is not showing. But okay, anyways. Initially, AI was used mainly to assist with image analysis. So as computing power and then the availability of medical data increased, researchers began developing algorithms that are capable of identifying tumor patterns that were often too subtle for the human eye. So more recently, AI has been moved beyond simply analyzing just images. And today, as we speak, it can actually assist with treatment planning, predict therapeutic response, and even support real-time clinical decision-making. Alongside this evolution, we have seen remarkable improvement in diagnostic performance. Deep learning algorithms now routinely achieve diagnostic accuracies above 90% for several cancers, including breast cancer and colorectal cancer. If you go further in the review paper that I published, actually, there are quite an extensive elaboration on this, and especially the deep learning algorithms like the convolutional neural networks and how it's integrated with other systems that was generated, and then their accuracies. There's a nice table, I think, that speaks on that. Okay. So this doesn't mean AI is replacing pathologists or radiologists. Instead, it actually provides an additional layer of analysis that can improve consistency, reduce variability, and then support faster clinical decisions. So one of the areas where AI had the greatest impact is medical imaging. I just want to show you, okay? So AI across optical imaging modalities. We have a tumor margin that is very unclear. But with the assistance of AI, you could see how enhanced it has been. So this is the fluorescence imaging. Okay, so it enhances tumor margin detection by reducing background noise and improving image segmentation. Okay, so this is just for you to appreciate what AI currently is doing in terms of imaging. We also have Raman imaging. Okay, so Raman spectroscopy provides molecular information, almost like a chemical fingerprint of the tissue. So you have a benign tissue, you have a malignant tissue, and this is the raw signal if you use the equipment or the tool. But with the help of AI, you are able to actually see clearly, distinctively, okay? Also, there's a tool, optical coherence topography, or OCT for short. It generates high-resolution cross-sectional images of tissue structure, as you can see here. Okay, the raw image, when you use OCT principle, this is the raw image of your tumor, and then AI enhanced. Clearly, you can see how it enhances the visibility. Okay, same applies with photoacoustic topography. Okay, so although these techniques differ, they all face similar challenges. Medical images can contain noise, poor contrast, and enormous amount of information. What AI is going to do is to help overcome these limitations by improving the image quality, which I've shown you already or demonstrated, enhance tumor detection, and also automate the image interpretation. So the important message is not that AI replaces these technologies. No, it rather makes each of them more accurate and clinically useful. So imaging is only one part of cancer treatment. The next step is delivering therapy more precisely, and this is where nanomedicine becomes particularly exciting. So earlier in my presentation, I showed you how nanoparticles can improve drug delivery. Okay, so artificial intelligence now offers an opportunity to make those nanoparticle systems even more smarter. AI can analyze enormous data sets to predict which nanoparticle designs are more likely to reach tumors effectively. Okay, it can optimize particle size, shape, surface chemistry, and drug loading even before the laboratory experiment begins. And it's a good thing because it pre-informs you, okay? So there will be minimal wastage and all that. So during treatment, AI can also predict how nanoparticles distribute within the body and even estimate the most effective light dose for activation. You don't just select any photosensitizer and any laser light. You need to know what you call their wavelength, all right, and then their surface plasmon because they need to match. Because, for instance, in this study, in the first part of the study, I used berberine. Berberine has a specific surface plasmon, which you saw, all right? And you cannot, and it falls within the blue light region of the electromagnetic spectrum. So you cannot use a laser that is around 600 to 800 nanometers to work on berberine, which falls within the blue region of 400. Okay, it will not excite it because we remember we need to excite the photosensitizer before we can get our ROS, our radioactive oxygen species. That is the only way we can kill the cancer cell. Okay, all right. So beyond treatment planning, AI is also accelerating the discovery of entirely new photosensitizers and nanomaterial. And that is also very important because we need as many of them as we can, okay, and try them and see which ones are more advantageous of the other. So instead of relying solely on trial and error experimentation, researchers can now use computational models to identify promising candidates much more efficiently. So in other words, AI is just helping transform nanoparticles from passive drug carriers into very intelligent therapeutic systems. Uh, Traditional medicine often follows a standardized approach. Two patients with the same diagnosis frequently receive very similar treatment. However, we know that cancers differ genetically, biologically, and clinically. Artificial intelligence enables us to move beyond this one-size-fits-all model, okay? So by combining imaging, genomic information, pathology, and clinical history, AI can actually help predict how an individual patient is likely to respond before treatment even begins. Okay, so it can optimize the photosensitizer dose, the laser parameters that's required, and treatment schedule for each patient. As new information becomes available during therapy, AI can continuously update these recommendations. So this represents a major shift from standardized medicine toward truly personalized precision oncology. So while these advances are exciting, important challenges remain that must there are certain challenges that really remain that must be addressed before AI becomes part of routine cancer care. Yeah, so at this point, it may sound as though artificial intelligence is ready to transform every hospital tomorrow. The reality is more nuanced, okay? Significant progress has been made, yes, but several important challenges still remains, okay? Like I always say, efficiency of a machine can never be 100%. First, AI requires large, diverse, and high-quality clinical data sets for training. Second, clinicians need to understand why AI reaches a particular recommendation. This concept is known as the explainability of AI, okay? And is essential for building trust, okay? Third, every new AI system must undergo rigorous clinical validation and satisfy regulatory requirements before widespread implementation. It is very important. Okay. And finally, successful adoption depends on integrating AI into existing healthcare systems without disrupting clinical workflows. So the future of AI in medicine actually depends not only on developing better algorithms, but also on ensuring that they are safe, transparent, and clinically reliable. Throughout this presentation, we've seen how artificial intelligence is already improving cancer imaging. I demonstrated for you, treatment planning, and nanomedicine. But this is only the beginning, okay? The future of cancer diagnostics will not depend on one technology alone. It will depend on the integration of AI, advanced imaging, nanotech, genomics, and clinical medicine into a single intelligent ecosystem. All these must be in a single intelligent ecosystem. So one exciting concept is what we call the digital twin. A digital twin is a virtual model of an individual patient that can stimulate how their tumor is likely to respond before treatment is even given. So this could help clinicians choose the most effective therapy while avoiding unnecessary side effects. Another emerging area is closed-loop phototherapy. Instead of delivering a fixed light dose, future systems could continuously monitor the tumor during treatment and automatically adjust the light dose based on the patient's real-time response. Okay, and that's actually also a good thing. We are also seeing rapid progress in generative AI, which has the potential to accelerate the discovery of new photosensitizers, nanoparticles, and treatment strategies by analyzing vast amounts of scientific data. Ultimately, the vision is a healthcare system where diagnosis, treatment, monitoring, and decision-making are seamlessly connected, allowing every patient to receive therapy tailored specific to their disease. I would like to leave you with five key messages from today's presentation. First, light-based cancer therapies have enormous potential because they allow treatment to be delivered more selectively, reducing damage to healthy tissues. Now, the second message is our experimental work demonstrated that combining green synthesized silver nanoparticles with berberine enhanced drug delivery. It also increased reactive oxygen species generation, the ROS, and also it promoted cancer cell death more effectively than the individual component alone. I demonstrated that to you. Another take home is artificial intelligence is rapidly transforming cancer diagnostics by improving image interpretation, supporting treatment planning, and enabling more personalized clinical decision. The fourth one I would like you to take home is the future lies in the convergence of nanotech, optical imaging, AI, and precision medicine. These technologies should not be viewed as competing approaches, but as complementary tools working together. And finally, while significant challenges remain, which includes data quality, explainability, and clinical validation, the progress being made gives us every reason to be optimistic about the future of precision oncology. If there's one message I hope you remember today, if there's one message I really hope you remember today, it is this: the future of cancer care is not just about developing better treatments. It is about developing smarter treatments that are tailored to each individual patient. As I come to the end of my presentation, I would like to gratefully acknowledge the various funding agencies and organizations whose financial support made this research possible. Their investment in scientific research continues to advance innovation and improve our understanding of cancer and its treatment. I would also like to extend my heartfelt appreciation to the director or the head of Laser Research Center, distinguished Professor Heidi Abrahamse, for her visionary leadership and for creating an outstanding research environment that encourages innovation, collaboration, and scientific excellence. My deepest gratitude also goes to my supervisor, Professor Blasan P. George, and my co-supervisor, Dr. Paramita Sabadikari, for their invaluable mentorship, guidance, and patience, and unwavering support throughout my master's research and my continued PhD journey. Their encouragement and constructive advice have actually been very instrumental in shaping me as a researcher. So to everyone who has joined this webinar today, thank you very much for your time, your attention, and your interest in my research. I sincerely hope that this presentation has provided insightful or has provided you with useful information and then existing opportunities at the intersection of cancer research, green nanotech, photodynamic therapy, and emerging role of AI in precision oncology. As you can appreciate, given the time available for today's webinar, it was actually not possible to cover every aspect of the research in detail, particularly the second part of my presentation, which is actually based on my recently published review article. Okay. The publication provides a much more comprehensive a discussion of the concepts, supporting evidence, current advances, and future perspectives in AI-driven cancer diagnostics. Therefore, if today's presentation has sparked your interest, or if you have questions that could not be addressed during the webinar, I warmly encourage you to read the paper for a more in-depth understanding of the subject. And finally, I would like to express my sincere appreciation to the organizers of this webinar for the kind invitation to present my work. My very good friend, Orwell, I'm very grateful. It has been a great honor and privilege to share my research with such a diverse audience. Once again, thank you to everyone who has contributed to this journey. And thank you for your kind attention.
Thank you very much, Ike. That's a fantastic presentation. Very beautiful presentation. Thank you so much. We are very much grateful. So please, the floor is open for questions. I'm sure some of you have a lot of questions to ask Mr. Baidoo, and I'm sure he's prepared to answer them. So
As for preparation.
if you would want to type your question in the chat box, you can do so. But if you also want to just speak directly, you can just show me your hand. I'll call your name, and then you ask your question. So please, the floor is open for questions. He spoke on nanomedicine. He spoke on the work that he's doing in relation to breast cancer, and then he also um, talked about precision oncology and then AI, that's artificial intelligence, combining them with nanomedicine, the pros, the cons, and all of that. So please, if you have a question, you can ask now. Okay, Augustina, please ask your question.
Okay. Please, I hope I'm being heard.
Yes, please, I can hear you.
Okay. Thank you for this beautiful presentation. I was following to see how healthy cells are being preserved. How did you preserve these healthy cells so that the rust could not destroy them?
Okay, thank you very much, Augustina. It's actually a very good question. My master's work was on 2D models, and it was focused on just the breast cancer cell lines. We didn't create any environment to mimic what is actually happening in the human body. Okay. And that is why I'm continuing this project. And that is one of the objectives that I'm pursuing right now. So I'm actually targeting 3D models, both healthy cells and non-healthy cells. And what I'm doing currently is more targeted, which means this time my nanoparticle, you you functionalize it with a compound that has the affinity, or it has the love for the tumor. You know, most tumors have antibodies that they express. There are certain things they express on the surface so that we all understand. So if you want the delivery to be more targeted, you need to now functionalize the nanoparticle, and then it moves directly towards the tumor, and then it has the ability to penetrate. Remember, this is just 2D for you to understand and appreciate what is going on, because we needed to know whether our nanoparticles would work before we proceed with actual work, like which are more clinically based. I hope I answered you.
Please, yes. Yes, I get it now. Thank you.
Thank you.
Right. Thank you very much, Augustina, for your question. Please, the floor is open for questions. Okay, Ruth, please ask your question.
Okay, thank you very much, Isaac, for that presentation. So, I'm a medical physicist, and I think my mind went quite wild with the whole presentation. But I don't know how radiobiology and then the 5 hours of radiobiology work with photodynamic therapy. And so, I don't know if my question will still be valid to your question, but I'm quite curious. How would fractionating the light dose affect the 5 hours of radiotherapy and then the immune activation?
Okay, thank you very much. That is quite an intensive
Yeah.
yeah, like okay, I need to actually consider, because I've not really done much with radio, I must
All right.
admit. Yeah, I
Okay.
must admit. So one thing that we do with laser here is, because we are working with nanoparticles that are metallic, we choose
Okay.
the laser power based on the surface plasmon of the metal.
Mm-hmm.
Because remember, the metal must contribute to killing the cancer cell,
Yes.
right? So it is not just serving as a delivery mechanism for the photosensitizer, itself is also contributing through photothermal activations, right? But I think I need to really look at the 5 hours of radio, and then maybe in future we can talk about it via email and then see how to go about it.
Yeah, thank you. I'll be very interested in that as well. Thank you, though.
Right.
Great
Yeah, I can do
presentation.
that for you.
Right. Ruth, thank you very much. Thank you very much for this question. I must admit, for some of us who know nothing about nano medicine, it's a lot. Anyway,
Oh, I understand you.
anyway, so please let me ask a question. The translational impact. So yes, you did 2D. You are hoping to do organoid cultures, if I understand it, that's the 3D, right?
Yeah, yeah,
So how for the layman,
right.
how will it happen? So let's say you are going for nanomedicine kind of treatment. Is
Mm.
it like they are going to inject you with the combination, the conjugate, and then they will then target it? Kindly explain that to our audience so that
Okay, okay.
yeah.
I get you. There are various aspects of nanomedicine. As of now, I want to publish a paper on microneedle insertions, all right? So typical, let me just calm down because of the mixed audience. For instance, the breast cancer, we intend to inject it directly at the breast region. We are not interested in swallowing, and then now it has to go through the lymphatics and what, and then there will be absorption. That is going to disrupt a lot, because remember, the blood has its own proteins and stuff like that. It would end up reacting with the nanoparticle. It will become more bulkier. It cannot move. Okay. So what photodynamic therapy or nanotech is doing is very precise. We target, if it is breast, it's breast. You inject it, and then it goes through, like I showed a picture of the mechanism of PDT. You see that there was blood vessels around the tumor that carry the photosensitizer. So that's the whole goal. Just inject it, and then it would use the EPR mechanism more like so that everybody understands, like osmosis. It would have the ability to just move from the blood into the neighboring tissue, and then it will be accumulated. It will not be able to go anywhere because of the targeted therapy, like I mentioned. That's what I'm doing for PhD. So you put certain molecules on top of it, which only has love for the tumor. So it will not select a healthy tissue, but it will select the tumor. All right. So like hyaluronic acid that has been conjugated onto the berberine silver nanoparticle. Hyaluronic acid has the ability or has the love for CD4 for cells. Okay. And CD4 for cells are highly expressed on MDA MB cells. So assuming your breast cancer is the MDA type, okay, which means that if I functionalize the nanoparticle with HA, it is not going anywhere, it is coming to it. Do you understand? So that is just for everybody to understand that we are dealing with targeted therapy now, and it's actually very promising. And I hope that in future, this whole tech would actually help save lives.
Please pardon my ignorance. So as soon as assuming you inject the conjugates, let's say in this case the berberine and then the silver,
And the silver nanoparticle, yeah.
it will just start working after you've done all the molecules, adding the HA and all of that. It will just start working without any laser.
Yes.
That's what it means.
No, so that's the thing. I didn't speak much on the chemotoxicity and then phototoxicity. Berberine itself has anti-cancer properties. Silver nanoparticle has anti-cancer. So there's synergy. Even without the light, these two combined has some lethality towards cancer cells. Do you understand? So that is what we call the dark toxicity or the chemotoxicity. So without the light, even just the two combined alone, what we will see that the cells are dying. And it showed that I had some very nice data. And once the work is published, you can read and understand properly. But what PDT seeks to achieve is killing the cells even faster and more. Remember, so now you've already introduced the conjugate into the breast tissue, which is the tumor tissue. You've introduced it, okay. Without even throwing the light, it is killing the cells. Now, if you throw the light on it, and then you see the effect even more massively happening. Do you understand?
Yeah. Yes.
So that's basically what is happening here. So it is good that you choose your photosensitizer wisely, and you choose your metal nanoparticles also wisely. I think I have a paper also on metal nanoparticles, and you can also read on that, and you will see there are various forms of metal nanoparticles. You have the gold, the silvers, magnesiums, and many others.
Please, my last question has to do with the reactive oxygen species. We know that they are radical, right? So they can also affect the person, naturally. So how do you, for lack of a better word, regulate the amount of reactive oxygen?
Yes.
Yes.
So you see, I did generating the ROS outside the cell. There's a data published on that. And then when the nanoparticle was introduced into the cell, I also measured the relative ROS intracellularly. Do you understand? If you check the values, they are not the same. So if you do it outside, it gives you a fair idea how the potential of whatever you are dealing with. So now you'll be measured. And remember, you do optimizations. You don't just get to where I got to. You optimize, and then you come out with a very fine concentration that you actually need. That will not be too lethal to kill even neighboring cells and even the human at large. Okay, so there are a lot of optimizations that you would do. You would calculate for the MTT assay would help you calculate for inhibitory concentrations. That is actually what you need to go forward with every other experiment. You need to know the inhibitory concentration at 50% before you carry on the experiment. Without it, you cannot, because you don't know what you are dealing with, how much concentration you actually need for you to even inject and even throw light on, and how much rust it is able to generate, and the impact of the amount of rust that you have. Okay, and that is why AI is also coming in, because all these things are done manually and it's hectic.
Very much. Thank you very much for answering.
Yeah.
I appreciate that. So please, any other question for Ike?
I hope there's none.
Suminwa, please ask your question.
Hello, everyone. Can you all hear me?
Yes, we can hear you.
Okay, great. I do agree. Thank you very much for the presentation. I do agree that I do not have any medical background as well. But my question is, you just mentioned that there are multiple metals that you could have used. Why specifically did you use silver nanoparticles? And as well as, I have another question, sorry. You said you used 2 cell lines. Why specifically did you use 2 cell lines when there are probably multiple as well? Those are my questions.
Thank you very much, Ms. Tuminwa. Considering that you don't have medical background and you are able to ask such a question, that's impressive, and it shows that you were following the presentation and you are overthinker. So that's good. I'll start with the silver nanoparticle. It has intrinsic antimicrobial and anti-cancer. So that is one. I mean, I want to see the synergy. So synergistic effect of my nanoparticle. So if there's a metal nanoparticle that already on its own can kill cancer cells, I think it will be nice for me to jump on. Okay, so that is one. And also, it is relatively less easy to synthesize silver nanoparticles than synthesizing gold. Okay, yeah. And then it is less expensive. Silver nanoparticles are less expensive to make. Even silver itself is less expensive compared to gold and other metals. Okay, and then in terms of, if I'm being more technical, the surface plasmon. Okay, considering that I'm using berberine, whose wavelength is around 466, 400, all right, that's the blue region. I need to select a metal nanoparticle that is also in the blue region. I cannot go for something that is outside the blue region. For instance, gold is outside the blue region. It's like 500 thereabout, which means that when I conjugate, there's going to be a problem with phototherapy, the photodynamic therapy. So considering that, I needed to use something that falls within the surface plasmons of berberine as well. I hope that answers you. The second question has to do with the 2 cell lines. Right. MCF. Okay, so this, like, biologically, they are distinct cell subtypes of breast cancer. Okay, MCF is hormone receptor positive, and then MDA is triple negative. It doesn't respond to hormones. Okay, it's very aggressive. That is MDA, and MCF is less aggressive. MDA is very invasive. It's quite wild. Okay, so testing both models allowed us to actually evaluate the formulation across the cancers with different characteristics. I mean, it only informs us that, okay, in both scenarios, what is our nano formulation doing? Worst case scenario, if you are dealing with a very aggressive cell line, what is going to be the fate of our nanoparticle? If you are dealing with a less aggressive one, what is going to be a fate? So you have a broader overview, broader idea. Okay. So I think that answers you.
Yes, it does. Thank you very much. And wonderful presentation again. Thank you.
Thank you.
Right. Thank you very much, Timinwa, for your question. Mr. Baidoo, there's a question from Wilfred. He says, "Hello, Isaac. That was a wonderful presentation. Are there any specific AI tools you know of that are being used currently for the predictive modeling you spoke about?"
Yes, I think if I remember correctly, there's the CNN, the convolutional neural networks. It has high accuracy image segmentation. So most of the deep learning algorithms are built around it. There are things that are being integrated there. And I remember in the review paper, quite a lot of people have published around it, and it's actually doing wonderful things. Like there's a softmax CNN classifier that had an integrating neighbor. That's the neighboring ensemble predictor. Okay, so it helped in accurate tissue characterization without requiring explicit segmentation. So yes, that is what I remember for now. I think, yeah.
Okay. Okay, so this is a question from Vincent. Wilfred has acknowledged your response. There's a
Thank
question
you.
from Vincent. He said, aside AI models, have you tried using packages to segment or create binaries for your images?
No, actually, I have not. I have not. And I must admit, this AI thing, I've not tried it. I only published a review paper because of the interest that I have and where precision medicine is going. So that is something I'm actually considering. I'm not an expert in using AI in PDT as of now, but it's an interest. And as like I've published a paper, it means I have something, I'm cooking up something. And please watch out. Thank you very much, Vincent.
Right. He has also acknowledged your response. And please, any other question? Any other question for Mr. Baidoo? Okay, so I believe this session has been insightful. It has been educative. We've all learned a lot. Vicente says he enjoyed your presentation and thumbs up. Thank you very much, everyone, for joining this particular session. And thank you so much, Mr. Baidoo, for making the time. I know you're a very busy person, but
Yo,
you made the time to present to us today on your research work and things that you are doing. And we are very much grateful. Our next session is next week, Friday. We are having Madam Augustina Ifia Santua Kwashi, who will be presenting on immunotherapeutic approaches in sarcoma management, emerging targeted translational insight. So these three sessions have been cancer neuroscience, nanomedicine, and then we'll look at immunotherapy in sarcoma management. I hope that you tell a friend to tell a friend to tell another friend.
right.
We can. And once again, Mr. Bailey, thank you so much. God bless
Yeah,
you.
please, can I add, can you put my email in the chat box so that if anyone wants to reach out for
Right.
further discussion, collaborations?
Kindly type it in yourself for me right now. I'll give you
Oh,
5 minutes. Yeah. So that.
okay. Let me. And even collaboration, if you want us to collaborate, we are open.
Right. That's great. That's great. Right. So I has placed this email address in the chat box. Please let's use a moment to copy if you want so that you reach out to him. Yeah. ibaidoo@ug.ac.za. Right. Okay, so I believe this has been an insightful presentation. Thank you very much for all of your questions. I'm really happy. I'm really happy that we had a fantastic session. Unless anyone has a question, I would like to bring the session to go. Probably a question has hit you right now, yeah. But if not, then thanks again, Ike, and then we'll bring the session to a close. So thank you very much, all of you, for making the time. See you on Friday. I hope to see you there, and then we'll have a wonderful session with Augustina. So Ike, have a blessed day, and all of you too have a blessed day. God be with you.
Right. Thank you all.
Bye-bye.
Bye.